Good afternoon to the Worldwide Neuro Theory Forum. Today I am very happy to present you with my friend Henning Sprekeler. But before I do, I would like to remind you to wear your seatbelts and to use at your leisure the chatting function on your right-hand side, as well as the ask a question function at the bottom of your screen. Now, today's feature will be presented to you by Professor Dr. Henning Sprekeler from the Technical University of Berlin. I met Henning when I studied physics with him in Berlin as well, 21 years ago. And since then, Henning has graduated from his physics degree, then gotten a doctoral title, also in Berlin, went on to become a postdoc in Wolfram Gerstner's lab, where I was fortunate enough to meet him again, and fortunate enough to collaborate with him for a good 2 years, before he left me and went back to Berlin with the Bernstein Prize, and then shortly after left for Cambridge, for the UK, where I joined him actually shortly after moved to Oxford. And while Henning is reconnecting, there you are, he shortly after, a year and a half afterwards, moved back to Berlin again. You can see he wasn't able to escape, where he's now a full professor in the computer science department of the Technical University of Berlin. And that's it. Without further ado, thank you very much for coming today, Henning. I'm looking forward to the talk.
Yeah. Thank you very much for the invitation. It's a great opportunity, and I'm very excited to present here. And so the first time I'm actually giving a talk on that type of forum, which is a bit daunting as well, because it's sort of a relatively large audience. One note up front, basically I can't see the chat window because of the stupid keynote grabbing the whole screen thing. So if there are any questions, Tim, then in between, that would be great if you could relay. Other than that, yeah, so what I'm going to talk about today is basically recent work that we've done on Interneuron circuits. And first of all, of course, it's the usual PI thing. I'm not the person who actually did all the work. The people who did the work are basically those people that I've listed here on the title. I will flash them more over the course of the talk so that you actually see their faces when you meet them at a conference at some point. It's great people to work with. So, what's this whole thing about? It's basically starting with this type of picture. So we've been brave enough, or maybe even stupid enough, to stare at these types of pictures over the last couple of years quite intensively. So what you're seeing here is sort of a cartoon of a cortical circuit. And all of you will recognize immediately the pyramidal cell in the middle with its nice little apical dendrites and basal dendrites. And the rest, all the colorful stuff, is interneurons, right? So these are inhibitory cells. And the thing that immediately strikes you is that this is a sort of a puzzling diversity of different cell types that have different morphologies, they have different electrophysiological properties, they have different targets, etc. And so what we've been sort of trying to do a little bit is to try to somehow get an idea of trying to understand what these things do and what this diversity actually means, right, or where it comes from. Now, that's not a trivial thing to do, but I think it's something that I think is actually sort of a cool place for modeling or for theory in general, because complex systems like that, you know, are hard to reason your way through. So it can be highly useful to actually have a computational model that helps you reason your way through these types of models. So the ultimate question that is, of course, here at stake is the question how the structure of these types of circuits is related to the function that the circuit performs, right? The idea is ultimately, of course, that this does something. This does some kind of cool information processing, but we just don't know why or how or which function. And so the traditional way of modeling these types of complex systems, or one of the traditional ways in computational neuroscience, would be bottom-up, right? Over the last decade or decades, basically after the advent of all the neat little optogenetic techniques, we've amassed a lot more knowledge about these different interneuron types, how they're connected, and potentially how they're related to function. So we feel that we can set up a model that just incorporates all of that knowledge and then sort of stare at that model for a long time and then see if we can infer what it does or what the respective function of the different cell types is. So that's something we've tried for a while. I can tell you that it wasn't fun. But there's also the other way of going about it, to basically start with a hypothesized function and then basically see if you can derive from that what the structure should look like, and then compare that to biology. And that's the approach that we followed much more over the last couple of years, and which, at least in our hands, has been more fruitful. So maybe to tell you roughly what the challenge here is, let's say we acted as if we would take that type of circuit and we'd reduce it to a compact model. And then I guess the starting point, would be, the pyramidal cell in the middle that might receive bottom-up input and sort of the perisomatic domain and top-down input on the dendrite. And then we could start adding interneurons, right? And so the way one would start to go about it, I guess, is to take those findings that are super robust across the literature. Right? So we could say, hey, the provalbumin positive interneurons, they target the soma. There are somatostatin interneurons, basically Martinotti cells and cells like that, which have the tendency to target the apical dendrite of these pyramidal cells. So that's, well, a good starting point. We know that the synapses onto these two types of interneurons have very different properties, right? So the synapses onto provalbumin positive cells are depressing, synapses onto somatostatin interneurons are facilitating. Okay, so that's good. But then there's more, right? There are other cell types. There could be VIP neurons, which seem to be in competition with somatostatin neurons. Somatostatin neurons inhibit all sorts of other cell types. But then there's, of course, more than just these three classes, more recent sort of additions to the zoo, or maybe NDNF positive cells that sort of Johannes Letzkos has characterized, that seem to also go onto the dendrite, but have a completely different time scale. So you'll see that this model relatively quickly grows in sort of complexity. So if you try to model that type of thing bottom up, there's an inherent problem, and that problem is simply parameters. And so let's just look at this figure here, where we have 4 different interneuron types and a pyramidal cell that gives you 5 cell types. If everybody were connected to everybody, we have 25 parameters just for the weights, and that's already collapsing each of these populations to basically a single population, forgetting about the identity of the interneurons. So in our hands, we've hammered our head against the wall trying to do a bottom-up model of that. Because even if there is beautiful data out there, even within the sort of the confines of what's possible within those sort of known parameters out there in the literature, we could basically make those circuits do whatever. Right? So that was kind of inconclusive, and that is sort of frustrating. So in our hands, at least, the bottom-up approach was hopeless. Obviously, I can't speak for others, right? There might be people who just know how to do that better. We couldn't really. So the alternative approach, as I said, that we took was to try to optimize for a given objective and then see if we can compare that to experiments. Now, at first glance, it feels as if we haven't gained anything, because obviously it's not so clear what that objective should be, because we don't know what the function of these circuits is. So we have to sort of think about what the objective could be. Now, when you think about that type of objective, there are different ways of going about it. You could either think about something like, if you wish, like an ultimate objective, right, which would be like a behavioral function or so. But that's difficult to guess, right, because it's not clear which behavior exactly we should take. So what we basically went for is something more like a proximal objective, right? So think about what the purpose of inhibition could be, and then see if we can shape those circuits such that they fulfill that function well. And one of the very sort of classic functions of inhibition is that of balancing excitation, right? Now, there's a very rich literature about what's known as excitation-inhibition balance, and I'll just flag a few. I think basically excitation inhibition is sort of one of the classical examples of beautiful theory-experiment interaction. So, for example, we know that excitation and inhibition are sort of balanced across different stimuli. So what you're seeing here is basically the tuning curve of excitatory and inhibitory currents onto a cell in primary auditory cortex. And I guess what you see is that there's a frequency tuning, so it's basically showing pure tones. And you see that both excitation and inhibition kind of prefer the same frequency, right? So both currents are maximal for the same frequency. Excitation and inhibition are also heavily correlated in time. And so this is from a paper from Milan Lampo's lab. They basically did all sorts of fancy tricks to try to record excitation and inhibition at the same time. And basically, you can see by eye that these things are correlated. when you look at sort of cells and check how much excitation inhibition different cells get, then it also looks as if excitation inhibition are correlated across different cells. This is from the Scandiani lab, which is sort of visual cortex, I think layer 2, 3, receiving input from layer 2, layer 4. But it's also sort of across cells, it's also correlated. Across patterns that you present, it's also correlated. This is very recent work, basically showing that if you do pattern stimulation in the hippocampus, that the postsynaptic excitatory and inhibitory currents are correlated. And then finally, very recent stuff is basically showing that if you just image how many excitatory and inhibitory synapses are distributed across dendrites, that you sort of have a local coordination of how many excitatory and how many inhibitory synapses you find there. So just to summarize, this basically a surprising sort of level of precision at which excitation and inhibition seem to be correlated across neurons, across stimuli, across time, across the neural morphology. And so this is something that we took sort of as a proxy for something to go for, right? Something to optimize. And surprisingly, we got comparatively far with that. So I can't tell you about all the stories, but it's sort of I'll sort of touch upon a few today. So we've basically found that if you just take a system in which excitation is learning and inhibition concurrently tries to balance the excitation through sort of means of inhibitory plasticity, you can explain spatial representations in the temporal lobe. You can get grid cells, place cells, things like that, depending on how selective the interneurons are. We found that if you optimize for excitation inhibition balance, you basically can provide sort of a reproduction of the fine scale circuit structure in mouse V1. You can basically balance excitation and inhibition, and what comes out is something like prediction error neurons in a setting where you have motor efference copies and sensory data. And you can also, even when you try to balance excitation and inhibition across different compartments of the pyramidal cell, you can actually just basically generate through the optimization different cell types, right? So you basically get this distinction between parvalbumin positive and somatostatin neurons, basically just out of the optimization. So what I'm aiming to do is basically to start out with talking about this part up here, which is work that is done by Owen Mackwood and Laura Naumann. Then I'll sort of move on to this part and then briefly touch upon this one. This is work from Loreen Herteg, and she'll present it actually at a workshop that we're organizing next week at the Bernstein conference. So it makes limited sense that I talk about that extensively because she's, of course, the person who did all this, so she should get the stage for that, right? I'll just sort of do a bit of a teaser at the end. So in a way, what I'll be doing is I won't be starting with many interneurons. I'll be starting with just one interneuron type, ramping up to 2, and then sort of having 3 at the end. That's sort of roughly the menu for today. So let's start out with this story up here. So this is work, as I already said, by Owen Mackwood and Laura Naumann. So 2 very talented people in the lab. And so what they have been sort of following up on is the question how precise feedback inhibition is in primary visual cortex of rodents, right? And there's a neat little sort of story behind this, and I think it's probably good if I give you a quick ramp up to that, so that you're all on the same page. So the underlying question is basically maybe that of stimulus-specific excitation inhibition balance, right? So this picture that I've shown you here before, where you know, excitation inhibition seem to be balanced across different stimuli. And one question that one could ask is where this comes from, right? And so it's in an earlier work together with Tim, actually, we suggested that this is something that arises from feed-forward inhibition, but then you actually have to sort of, you know, find means to coordinate the 2 currents so that, you know, excitation inhibition arrives at different synapses. So you somehow, if you want to get those in register, you'll have to do something actively, right? But there's actually a different suggestion, and that suggestion is based on the very original suggestion of how EI balance actually arises, right? And that's sort of the work from Karl von Vreeswijk and from Polinsky, etc. It's sort of the idea that if you have just excitatory and inhibitory neurons in a random network, and you just make sure that this negative feedback loop here from the excitatory cells to the inhibitory cells and back is stronger than a potential excitatory feedback loop here on the excitatory cells, which I was not smart enough to draw, then you more or less automatically get that balanced state. And the logic is very simple. If you plot basically the input currents to this pyramidal cell as a function of the firing rate of those cells, then you basically see that as, sorry, that as you basically increase the firing rate, the excitatory input would go up, right, due to recurrent excitation that, as I said, I wasn't smart enough to draw. But the inhibition would also go up because you basically get this negative feedback loop here through the interneurons. And to be stable, this negative feedback loop should be stronger than the positive feedback loop. And so you basically have a situation where as the excitatory rate goes up, eventually the inhibitory currents will dominate. Right? And so in such a setting, if all of these currents are strong, then you're most likely to end up somewhere here in this regime where those two currents cross. And that's basically the regime where you're in a balanced state. Now, if you increase the input to that system, right, by means of stimuli, then what would happen is that this red curve here would just shift up or down. That would change the point where these two lines intersect, and that would change the firing rate that the cells show, but it would not change the fact that you're at this balancing point. Right? So with that idea, you could basically, in such a network, generate a stimulus-specific balance of excitation and inhibition without any need for fine-tuning, right? It just falls out. And so that's sort of the idea that Ken Harris and Tom Osage-Flogel basically pushed in a beautiful review in 2013, that this might just be the consequence, or that this excitation inhibition balance might just be the consequence of local connectivity in a system that has a topographic map, right? Because then locally, if you look around this topographic map, then locally, you know, all the neurons more or less have roughly the same tuning, so it would resemble something like a random network, and you could basically use that to explain that type of tuning. Now, there's a corollary from that, and that corollary is that if you lose the topography, that the system should look different. And that's sort of the idea that if you have this sort of topography, okay, everything locally is the same, but if you move to a system like, for example, rodent V1, where it looks as if there is no sort of clear-cut organization of the neurons, no clear-cut topography, for example, stimulus features like orientation, then locally, you would have a large group of neurons that all have different tuning. So if you'd pool over those, you'd basically lose the tuning altogether. And there would be no means of having sort of, you know, a specific feedback inhibition onto the different cells. And so this is something that is sort of at first glance what you would expect to happen, because the connectivity between excitatory and inhibitory cells is just extremely high and seems to be apparently random and sort of uncorrelated to what their tuning is. Okay, so this is basically a plot where you have was basically the difference in the orientation tuning of two cells, and it's plotting the connection probability between those different cell types. And you see that connection probability, at least for the, is pretty high. And it doesn't seem to depend very much on whether the cells have the same tuning or not. Right? So this is work from Sonja Hofer's lab. So basically, the idea would then be that, you know, interneurons pool over all sorts of different excitatory cells and thereby completely wash out any type of tuning because they're just averaging over everything in the surround. And so this is something that is consistent with what has been found, that interneurons in the visual cortex of mice have sort of a much less pronounced orientation tuning than, for example, excitatory cells. However, they don't have no orientation tuning. Right? So there is some degree of tuning towards orientation. Not sure if all of you are familiar with these sorts of polar plots. The idea is that along sort of the angle, you plot sort of the orientation of, say, a grating, and then in the radius shows you how active these cells are. So if this is a circular, then it's an untuned cell. If there's some deviations from the circle, then there's some kind of basic tuning that is present. The question is this just noise, right? Or is this something that is actually used by the circuit? And so what it looks like is as if it's actually used by the circuit. So there's all sorts of prior work that shows that if you look in excitatory cells and you plot the excitatory and the inhibitory conductance that these cells receive and plot it as a function of orientation, that there is actually this type of co-tuning as well, right? And this is something that's difficult to reconcile with the idea that, you know, at least with the idea of feedback inhibition from inhibitory cells that have no orientation tuning. And sort of recent work that is sort of, that I think is very nice is from Peter Znamensky, is the idea that if you basically look at PV interneurons, or what he did is he looked at PV interneurons in visual cortex very precisely and plotted not only orientation tuning, but also tuning to spatial frequency and temporal frequency, and found that these cells often have actually quite a pronounced tuning, at least in those axes, and that this tuning is actually predictive for how these cells are connected to the excitatory cells. So basically, if you look at how similar the responses of a given excitatory cell and a given PV interneuron are, which is this total response similarity, then it's a predictor for how strong the inhibitory connection from the interneuron to the excitatory cell is. And similarly, there's a quite not as good, but still a predictive power in that response similarity also in the opposite direction. So you can try to predict the EPSP from the excitatory cell onto the PV interneuron. And finally, if you have cells that are bidirectionally connected, you actually see sort of a correlation between how strong the connection from the excitatory cell is to the inhibitory cell and how strong the connection is on the way back. Okay? So if you put this back into this sort of graphic schematic, then then this picture is probably not really the more accurate one, but it should probably look more like this, where you have cells that have similar tuning that are sort of preferentially connected. And this is something that is sort of this might be loose, right? This might not be very precise, but it's something that is sort of reminiscent of something like assemblies that consist of excitatory-inhibitory cells that have similar tuning. And so the question that we're asking here is how this is actually achieved, right? How that develops, right? So it feels like there's some kind of plasticity that should be establishing that is activity dependent. So how can you get that? So the question is, of course, so the starting point, as I mentioned, was basically the idea of achieving a balance of excitation and inhibition. And this is something that Tim and I had actually been working on a good 10 years ago now, but we looked at it primarily in feedforward networks. And so if you have a cell that receives excitatory input, say from 8 different populations, an inhibitory input from say 8 different populations, and you wire the whole thing up such that excitatory input is tuned to which population is active, whereas inhibitory input is not, then you can go ahead and put plasticity onto these synapses here and basically use a very simple rule to basically establish a stimulus-specific balance. And the rule goes as follows, that you simply say, okay, let's check how active that neuron is. If it's more active than some kind of parameter, which we call the target rate, then you potentiate the inhibitory synapse. Right? And you do that not for all synapses, but only for those that were actually activated, right? Where there's presynaptic activity. And if you do that, then you relatively quickly get a scenario where excitation and inhibition are perfectly balanced. And the rationale is very simple. What this rule tries to do is it tries to get this firing rate to be exactly at target, right? Then plasticity stops. This target is a constant thing, right? So if excitation is something that's not constant, then the only way to achieve that constancy is to subtract an amount of inhibition that exactly reflects the variations in the excitation, right? So you basically need a perfect positive correlation between excitation and inhibition to do that, and that's exactly what you see here. Right? So how do we translate that to recurrent networks? Obviously, sort of just coming up with a rule is something one could do. But the nice thing is that if you look at this rule, it's actually something that you can derive from an optimization principle, right? And that optimization principle is simply to say, postsynaptic rate deviates from a target. That's not good. Let's square that average over the time and punish that, right? Basically, it's like trying to get the firing rate to be the target rate at all moments in time by means of minimizing mean squared error. And the important point here is the order of the averaging. You don't average here, right? But you average over the square. And that basically means that you try to push the firing rate to the target rate at every moment in time, rather than on average, which would be the traditional way of doing homeostatic plasticity, for example. So what this tries to do is aims to establish an EI balance at all moments in time, or sort of, for all stimuli. So the nice thing is if you have this as an optimization problem, you can do that pretty much in any kind of system. Right? And so this is basically what Owen and Laura did, right? Basically, they went ahead and took a recurrent network of excitatory and inhibitory cells. It's a rate network to keep things simple. The excitatory cells are given a stimulus selectivity. And that stimulus selectivity is, again, a little bit is akin to the things that Peter Znamensky did. So there's a spatial frequency, there's a temporal frequency, there's orientation. So these cells have an identity. And the interneurons basically in the beginning are just, randomly connected. And what you can then do is to say, hey, let's take that deviation from the target rate and minimize that. And the way to minimize that is basically just by gradient descent. And so we try to write down learning rules that basically minimize this objective. Now, this is a recurrent setting, and the aficionados among you will know that what you have to do there is basically backpropagation through time, and the update rules that you're going to get there are very non-local. Right? So this is something that's really not biologically very plausible, but it's something that one could in principle do. Now, I'll just briefly touch on that we've actually developed sort of a local and online approximation of that scheme, and that is actually comparatively simple in nature. Right? So if you do that, then what you get, even in the recurrent setting, is on the connections from the interneurons to the excitatory cells, exactly the learning rule that Tim and I suggested at the time. For the other synapses, the ones that go onto the interneurons, you can basically come up with a different type of rule that is still quite similar in nature. It doesn't compare the postsynaptic firing rate of that interneuron, but it compares the overall excitatory input that the cell receives from the pyramidal cells to, well, a scaled version of this target firing rate. And then you multiply that with presynaptic activity, and what you're ending up with is a rule that is basically local. And if anybody wants to discuss that even further, it's basically something that's very closely related to feedback alignment, where you basically replace the back projecting matrix, or these weights here that you would normally use for back propagation of error, you replace them by the feed forward weights, right? And those are positively correlated, as it turns out, so that this basically allows you to do an approximation of the gradient. Now, having said that, all that I'm going to tell you about is something that is basically in both the approximation and in the full gradient descent rule, basically indistinguishable. Okay? So let's see what happens when you do that. So the first thing that one could note is that you could check is whether these interneurons actually develop some kind of stimulus tuning. And it turns out they do. Right? So if you look at basically the orientation tuning of those cells, and you look at how they're tuned to spatial and temporal frequency, you see that they develop these types of blobs, so that the PV interneurons actually develop sort of a functional identity. And you can also do this statistically. These are just sort of 3 example cells. But if you look at the selectivity of these neurons, then initially in the random network, there's nothing, but that basically develops over the course of this optimization. Note that the selectivity is not as pronounced as it is for the pyramidal cells, but there is a substantial selectivity. Something maybe for those people that have looked at this data quite a bit, the measure of selectivity that we use here is actually skewness of the firing distribution across the stimuli. So that basically discards a baseline firing rate, which otherwise would very heavily change the measure of selectivity, such as orientation selectivity indices or so. Okay, so the next thing, of course, is to think whether this connectivity between the excitatory inhibitory cells is reproduced. And so we find exactly the things that Peter has found. So you basically see positive correlations between the similarity in these tuning properties and the inhibitory weight. We see sort of a little bit weaker correlations among this response similarity and the excitatory weight from the excitatory cells onto the interneurons. And basically, in bidirectionally connected pairs, we also see a positive correlation between the excitatory and the inhibitory weight. So we very much get sort of a circuit structure that resembles that of excitatory and inhibitory circuits in V1. So basically, something that's akin to excitatory and inhibitory assemblies in sort of a self-organized way by means of local rules. Now, of course, you can ask, is all of this necessary? And that's one of the advantages, of course, of modeling, is that you can afterwards do to that system whatever you want. Right? And so what we basically then did is we knocked out plasticity. Right? So you could say, hey, is it really necessary to have these synapses plastic? Is it necessary to have these synapses plastic? So we can just kill forms of plasticity and see how that affects the results. And so let's start out maybe with the situation that we had before. Both of these synapses here were plastic. But let's first knock out the connection, the plasticity, not the connections, right? The plasticity of the connections from the interneurons onto the excitatory cells. Now, if you do that, what happens is that, first of all, you see that these interneurons actually lose a substantial amount of their selectivity. So they're a lot more broadly tuned afterwards. And this is something that might, due to good old color map issues, it's probably not as clearly seen here, but it's actually not a lot more than for random. What you also lose is these positive correlations, at least between the inhibitory weight and response similarity. Now, some of you may say this is something trivial, but it's actually something that is not clear, because in that recurrent circuitry, although these synapses are now static, right, it could still be that the tuning properties change because of this plasticity here, and that you can see a correlation here. And actually, if you look at sort of a very, very large sample of thousands and thousands of synapses, you actually see a weak correlation that is basically just not seen when you're talking about very few synapses. So what we try to do is then also to see if we can compare this type of data to the experimental data. So we basically sampled a compared, sort of a similar number of synapses, of connections from our network, and then basically checked whether we find positive correlations for each of these three types of correlations. And so what is basically plotted here is that before the plasticity, there's very little chance of seeing a correlation in any of these three sort of scatters. When both are plastic, all three are significant. And if you knock out the interneuron to a pyramidal cell plasticity, then basically you see only one of these to be positively correlated, which is this one, and all the other ones are not. So that basically is suggestive that this plasticity is actually required to explain the data. We can also obviously knock out the other synapses, right, and see what happens then. And again, selectivity of the interneurons is basically gone. Now, this is mediocrely surprising because the interneurons are now randomly pooling over the activity of the excitatory cells, so we wouldn't expect much selectivity there. And this is actually, if you quantify this more, so if you plot the PD selectivity for these 4 different cases, say before plasticity, when both synapse types are plastic, or when either of the two are knocked out, then the selectivity is consistently reduced. And most so if you block this form of plasticity. Again, you can check how consistent the data are. I'm not going to show you the scatters now with the experimental data, and they're basically also not consistent. So the take home here is basically that we would expect that both of these types of synapses actually need to be plastic in order for to explain the types of data that Peter Znamensky has seen. So to explain that sort of notion of excitatory-inhibitory assemblies. Now, this is something that relates to work that was done, for example, by Ashok, Lidwin Kumar, which is sort of a very nice sort of law of large number argument, namely that if you have a comparatively large number of connections onto an interneuron, then basically this pooling here is something that is sort of subject to a law of large number. So if you don't have plasticity here, then these synapses, these neurons will basically not develop any selectivity. And without selectivity, it's very hard to basically get them functionally involved in a stimulus-specific way, right? And so there's basically an argument over law of large numbers here, which is, something that one could discuss otherwise and for time reasons. Okay, so interim summary. So basically, if we optimize these sort of recurrent circuits for a detailed EI balance, meaning a stimulus-specific EI balance, we basically get out a structure that looks roughly like principal cell PV interneuron connectivity in mouse V1. What I haven't shown you in detail is that this actually leads to a co-tuning of excitation and inhibition across stimuli. And so what I've more focused on is the fact that if you knock out either of these two forms of plasticity in that circuit, it's very hard to get these types of data. Now, what I haven't talked about is something that you can find in the paper if you want to, is that you get that type of thing, these EI assemblies, you basically get within these assemblies an inhibition dominance, right? And that is sort of related to work from Chris Harvey's lab from just last year, which shows that if you stimulate an excitatory cell, that you tend to suppress cells that have a similar stimulus tuning. But that's something that's in the preprint. Good.
Henning, we have 2 questions.
That's the preprint. Yes?
Yeah, first question is whether it's necessarily exclusive or if it's rather interneuron plasticity per se being critical. I'm not quite sure I understand the question, unfortunately. Also, I have large feedback from your speakers. But I suppose the question is whether it's critical that you see both types of these plastic effects because you switch all other interneurons off in your setting. Or in other words, what's the role of the inhibitory neurons that you're not modeling?
The other cell types in there. I mean, of course, it is in principle possible that it's possible that the tuning of the PV cells is carved out by basically, or it arises from other sources, right? I mean, it could be that basically they become tuned because, you know, there's a different inhibitory cell type that then shapes the response pattern of that inhibitory cell, which then generates certain types of correlations. But I'm not sure if that is something that would, you know, leave a trace necessarily on these connections, right? So if you look at the connectivity between the principal cells and the PV interneurons, then it would be hard to see how, if they're not plastic, you would see these correlations arising because other interneurons sort of shape that. But, I mean, I can imagine one could conjure something up, but I wouldn't immediately know how to do that. Did that answer the question or not?
I guess so. Francis, if you want to ask a follow-up question to read, I'll be glad to do that. In the meantime, maybe, Elif, do you want to ask your question?
Sure. Hi, Henning. How are things? Interesting stuff. So I'm curious about whether a salt and pepper organization versus a orientation maps organization of the visual system, for example, for mouse or for cat, would bring about differences in the orientation gene that you expect in the inhibitory cells based on your rules.
I think in the end, as soon as you move to a topographic setting, I would expect that the plasticity doesn't do much, right? Sort of, at least when you talk only about orientation, right? I mean, of course, the question is, of course, according to which stimulus features are you talking about where there's a topography, right? If you talk only about orientation and there's a topography in orientation, then local connectivity does everything for you, right? Because the interneurons then just, if they locally pool over the surrounding excitatory cells, and they all have the same tuning. And so, of course, they're going to inherit that tuning. So if you would go ahead and like come up with a plasticity rule that tries to generate EI assemblies, but these EI assemblies are already there, then there's really not much to do. Right? So in that sense, I think this is something. So it's more relevant for stimulus features for which there is no or a poor topography, right? So in that sense, there could still be features like that, even in, you know, carnivore visual cortex that is sort of, you know, topographically organized with respect to orientation. When you look at other stimulus features, you know, whatever spatial frequencies or temporal frequencies, and so I don't know which stimulus features are not topographically organized, right? But if you look at those, then maybe there is something to be found there that has an analog.
But so the experimental data you're comparing against is for a salt and pepper organized visual cortex, and what experimentally is known about the differences in terms of orientation tuning of the interneurons?
Yeah, I mean, it's less strong in the salt and pepper setting, but it's not gone. So I think that's basically the way I'd probably see it. So if you look at, I guess
the issue we face is, like, how do we exclude the null hypothesis that orientation is just due to inhomogeneities in an averaging of the salt and pepper?
Yeah, it's very hard to exclude that, and it's something we wondered about that because that's basically something that is related to the question whether law of large number applies or not, right? So if you are a specific interneuron and you're sitting there and you're pooling over the local surround, then the question is, how many of these cells are there, right? And not only that, sort of, it's also the question, how broadly tuned are those cells around you, right? If you have 100 cells and they all, you know, respond in 0.001% of all the stim for 0.001.
Did we lose Henning?
Did we just lose our speaker? Elif, can you still hear me?
I can still hear you, yeah.
So we lost Henning. Lovely. How you doing, Elif?
Pretty good, Professor Dr.
For your postdoc positions while Henning is scrambling hectically to come back online?
Yeah, we're hiring in Montreal. Please get in touch. Architectures of Biological Learning Lab.
Do you have a talk ready?
Not with results, but with general ideas, yeah.
Let's see. You might have to continue for Henning. Half his talk was done, so, you know. Oh, Henning can hear us. Great. But he's not coming back. Are you coming back, Henning? Well, let's say something entertaining for the audience, please. What are you going to start working on?
So our lab is called Architectures of Biological Learning. We're associated with the Mila Artificial Intelligence Institute. We're planning so it's going to be a sort of a neuroscience and AI mix where we're trying to, I spent the last 10 years at the Blue Brain Project. In our hands, we could build bottom-up models. That's maybe something interesting I could say of these interneuron systems. But it's a big, complicated network, and I do agree with Henning that theory is super important to trying to understand what's going on in these things, and trying to figure out which of these details are important and which of them are just sort of implementation details. And so we're looking at also pairing that with artificial intelligence, deep learning networks, convolutional networks, and trying to study learning rules.
Well, thank you very much. That concludes our interlude, and we'll be back to the main feature. Thank you very much.
So am I back? Can you hear me?
Yes, you're back.
That is good. When exactly did I leave?
Just in time in the middle of the conversation.
So I just managed to escape the question, or?
No, I think the question is all right.
Yeah, I think you did a pretty good job of answering in general. I think I got the general idea. So
okay.
Yeah,
why don't you move on since we lost a bit of time?
Okay, I'll move on to the next, because I think otherwise probably time will at some point start to be pressing. Very sorry about that. I have no idea what's going on here, but it's yeah. So.
And maybe I'll duck out.
Yeah. Okay. So yeah, if you want to learn more, have a look at Owen's and Laura's preprint. It's out there. Good. So that's basically the first story. The second one, I'll try to be comparatively fast. Is basically about the question how we can get different interneuron classes just from optimizing for excitation-inhibition balance. And so this is work that's primarily done by Joram Kaiser, a PhD student in the lab, doing great work. And it is heavily based on stuff that Richard Naud did in the lab when we had the luxury to work with him. He's now obviously moved on to PI status. So the whole point here is if you think back of all these different figures for excitation and inhibition coordination, the one that I'll be sort of focusing on now is this one over here, where you basically have something like a coordination of excitation and inhibition across different compartments of a given cell. And so so the question is a little bit what happens when you do that, or if you try to do that, and what that does to the inhibitory circuitry. So let me quickly start out. Obviously, we're doing this in a lot simpler way. We're not doing compartmental neuron models, but we're starting with sort of a comparatively simple two-compartment model that Richard at some point developed. So it's a two-compartment model of a pyramidal cell, but it is spiking, and it receives sort of bottom-up input on the soma and top-down input on the apical dendrite. And so what we basically do, or what Joram does, is basically takes a population of those neurons. And then takes a population of interneurons. They're completely undiversified. They're basically all the same. They're integrated 5 neurons. He provides the principal cell output to these interneurons, and they are allowed to sort of provide feedback inhibition to both the somatic and the dendritic compartment. Right? And initially, everything is random. The interneurons basically don't distinguish between soma and dendrite, etc. And then we go ahead and we optimize this basically for excitation inhibition balance, both in the soma and in the dendrite. Okay. Now, an important point here is that we not only optimize these weights here, but we also optimize the short-term plasticity of these connections from the excitatory cells to the interneurons, because that's sort of a prominent feature of these different interneurons, that this short-term plasticity is actually different for different cell types. And so, to be a little bit more precise, many of you will know that synapses have a history dependence, right? So if you have a spike and there's another spike right afterwards, it can well be that the succeeding spike actually induces a lower EPSP or EPSC than the previous one. That's called short-term depression. And then there's also the possibility that the next spikes actually are sort of strengthened, right? And that's called short-term facilitation. And that's sort of a standard model out there in the field, which goes back to Henry Markram and Misha Zudix. And so I won't explain the equations in detail. The main point is that there is one central parameter, which is this initial release probability. And that parameter has a strong impact on whether these synapses or whether these model synapses are facilitating or depressing, right? So if release probability is low, the synapses tend to facilitate. If it's high, then they tend to depress. And so what Johann basically does then is he basically optimizes the circuit, right, to minimize the mean squared error between excitation and inhibition in both the soma and the dendrite. And the parameters that are optimized are the weights from the interneurons onto the excitatory cells and this initial release probability for the input synapses onto the inhibitory cells. Okay. Now, a little bit of technical detail. This is basically done by backprop through time, right? It's a recurrent network, so you basically have to backprop in time at that point, because really this short-term plasticity is something that now leaves a memory trace that you have to propagate through. And so the way we do that is, good old automatic differentiation, so the sort of modern machine learning tools. And because everything is spiking, we actually have to do that with surrogate gradients, right? Because there's the spiking mechanism would normally not be differentiable. So that's sort of a little bit, sort of a touch on what's under the hood. For more details, Johann can tell you. The more interesting part is maybe what happens, right? And so what I'm showing you here is the initial condition before we actually start the optimization. So you have this population of interneurons, and you can plot how strongly every neuron projects to the soma and how strongly it projects to the dendrite, right? And this is sort of a disorganized cloud reflecting the fact that we initialize these cells randomly. Right? The color of these dots actually represents the property of the incoming synapses. Okay, so basically, this is sort of the color bar here. So PPR is the paired pulse ratio. It tells you how much, if the succeeding EPSP is larger or smaller. So basically, if this is a number above 1, then you have a facilitating synapse. If it's below 1, then you have a depressing synapse. Right? And initially, so we initialize all of these synapses such that the paired pulse ratio is 1. So they're kind of neither nor. Okay, and then you go ahead, or Johann goes ahead and optimize all of this. And what you end up with is a picture that is very different. So you end up with basically a population of cells that has virtually no projections to the soma, right, or very weak connections to the soma, but a substantial synaptic weight onto the dendrite. And a second population that is along this axis here, which is a very weak projection to the dendrite, but a comparatively strong projection to the soma. So we basically get a diversification in function of where these cells project, right? And the other thing that is probably worth noting here is that all those cells here tend to be yellow or green, right? So they're all receiving facilitating synapses, whereas these guys here are all blue up to dark blue, so they have to receive depressing synapses. And if you do sort of a Gaussian mixture model clustering of these cells, and then afterwards check, color them, then you see that the paired pulse ratio is actually bimodal, right? And that the cells that project to the dendrite, which is this class here, are the ones that have a positive, are facilitating, right? Have a positive or the paired pulse ratio larger than 1. So in the end, what that suggests is that you end up with a circuit that looks more like this, right? So you have a cell type, which is this blue cell type here that projects to the soma and receives depressing synapses. And that's very much the hallmark of basket cells, of albumin positive interneurons. And the second cell class here that projects primarily to the dendrite, that's this yellow cloud up here, that receives primarily facilitating synapses. And that's very much what Martinotti cells or somatostatin positive cells receive. So I think it's an interesting fact that you actually get cell diversity just out of optimizing balance of excitation and inhibition. But the obvious question is of course, how does that work? So I'll skip that in the interest of time. So I could show you that excitation and inhibition are actually balanced, but I think that'll become clear anyway. So there's sort of an interesting problem here. And the problem is the following, that you have these pyramidal cells, and they receive excitation on the soma, they receive excitation on the dendrite, and what comes out is something that's sort of a mixture of the two. Right? So the problem is now that what you would want to do is you would want to provide inhibition to the dendrite that is related to the excitation that the dendrite receives, and inhibition to the soma that's reflective of the excitation that the soma receives. So what you need to do is you need to, from this sort of mixed signal, this green thing, recover both of the inputs that were actually arriving on that cell. Right? So, and that's sort of a non-trivial decoding problem to think about. And so this is now the point where Richard Naud's work comes in and sort of comes to the physiological properties of these pyramidal cells. So many of you will probably have heard about this. So basically, when you look at these types of neurons in slice, and Matthew Larkham has done like sort of amazing patching experiments there, then you see heavily sort of nonlinear integration in those cells. So if you would do a somatic stimulation that is comparatively weak, then you tend to get a somatic spike. There is a certain amount of after hyperpolarization, but you don't get sort of a very pronounced effect in the dendrite. If you go ahead, however, and stimulate in the soma and generate a spike, then that spike can backpropagate into the dendrite. And if it's paired there with, sort of EPSPs arriving in the apical dendrite, then suddenly you get this sort of ping pong between soma and dendrite. You activate voltage-dependent calcium channels here in this apical initial segment, and you get a very pronounced and long-lasting dendritic plateau potential that generates a burst of action potentials. Okay, and so the question is, of course, what does that type of thing do? The traditional notion is that this is sort of a gain control. Right? So if you have top-down input, that your representation of the bottom-up input is boosted. Right? But there's also a different notion, and that's what Richard suggested. And this notion roughly works as follows, namely that if you have a population of those cells, then you can basically assume that they encode both of these signals at the same time, both the bottom-up and the top-down signal, in sort of a multiplex timing code. And the idea works as follows, namely that if you have somatic input, then this will generate spikes. Okay? These spikes can now basically be converted into bursts if there is top-down input. And if you assume that this conversion is actually something that's stochastic, then you would, within a population of cells, see some of them bursting and others not. And how many of them burst is then reflective of the top-down input that is arriving in that population of cells. Okay, so the prediction would basically be that you could basically decode the somatic input from something that we call events. Right, so if you don't distinguish between spikes or bursts, then you'd have that initial thing that you need, which is the somatically generated spike, and that is reflective of somatic input. Whereas dendritic input could be decoded by checking what the fraction of of events are that have been converted to bursts. Okay? And so this is sort of an idea of a multiplexing or multiplex code that arises sort of from basic notions of just integration of electrophysiological properties of these cells. So if that's the case, then there's basically a way of interpreting the interneuron circuit as a decoder. Okay? So the notion would be that you'd have excitatory inputs to the dendrite, excitatory inputs to the soma, that generates activity that leaves the cell. And now what you can try to do is you can try to just count events, right? And that would tell you what the somatic input was, and you could feed selective inhibition back to the soma, or you could go ahead and try to calculate sort of the proportion of events that were bursts in the population, and then basically feed that back to the dendrite. Right? And the suggestion here is that this is very much what's happening in this interneuron circuit and gives rise to this diversification. Okay? So let me just quickly walk you through that. If you first, that in this population, these cells actually work like that. So if you plot as a function of the excitatory input to the soma, the event rate of those cells, so not distinguish between excitation, between spikes and bursts, then you see that this is actually a comparatively good predictor. Right? So you could infer the excitatory input from the event rate. Similarly, as a function of the excitation to the dendrite, you plot the burst probability, meaning the fraction of the neurons that were active that were actually bursting. Then you also see that this is something that allows you to infer the dendritic input. Now, if you would want to calculate these quantities by biophysical means, then calculating the event rate would mean don't distinguish between spikes and bursts. Right? And the smartest way of doing that is to treat a burst as a single spike. That means you should just depress away any further spikes that arrive after the first one. Right? And that's something that can very easily be done by short-term depression. If you wanted to detect bursts, right, to calculate something like the burst probability, then you would need facilitating synapses, because you should throw away the first spike and only pay attention to any spikes that come later. Right? So this is something that you could very easily do with, or very easy, well, you can do it with facilitating synapses. And that's sort of the picture that that we're drawing here. Basically, that if you plot as a function of the burst probability, the firing rate of these cells here, right, that have these facilitating synapses, then you see that there's a nice positive correlation here. Joram can tell you about these error bars. If you plot as a function of the event rate of these cells, the PV rate, then you see that that is also nicely predicted. So it very much suggests that these interneurons are actually really decoders of that input that is arriving in the soma and the dendrite. Good. And that's sort of the final puzzle piece, basically, that excitation and inhibition in the two compartments are afterwards positively correlated. Right? Okay, so I think in the interest of time, I should probably skip more details here. Tim, how much time do you still give me? We started late, but I think I'm out of time, right?
No. We started 10 minutes late. So you still have at least another 10. And,
10? Really?
Then you have as long as you want. People are going to vote with their feet. Once we lose 50% of our audience, I'll cut you off.
Yeah. Okay, good. So so I think last part is really just briefly touching. As I said, it's basically just a sort of a teaser almost. So what I've talked about now is basically just sort of showing you in the first part that you can explain sort of fine scale structure in V1. The second part is this interneuron diversity in terms of somatostatin and PV positive interneurons that you can get from optimizing for dendrite and soma specific balance. And the final part is work from Loreen Hertag, an amazing postdoc in the lab who's sort of been relating these this excitation inhibition balance thing, which is sort of my standard meal, to prediction error coding, which she's done very, very nice stuff on. So how does that roughly work? And I'll only give you a teaser because, as I said earlier, Loreen will give a talk next week on Tuesday in our workshop where we talk about sort of the making and breaking of excitation inhibition balance. And I think we actually have a very nice lineup of speakers. And so I encourage all of you to attend. I think it's going to be a lot of fun. And so I'll only tease this quickly, and then I'll leave it to Loreen to explain this in detail. So prediction errors are something that is sort of or prediction error codes are something that is sort of quite broadly found throughout the brain. Many of you probably know sort of the good old dopaminergic signals as reward prediction error stories from Wolfram Schultz and many others. But prediction errors actually arise in quite a lot of other situations, and that's functionally something that's very useful, because if you think about how you would normally behave in the world, then you know these types of prediction errors are actually quite important. So something that you might actually all know is that the majority of the sensory input that we receive is actually thrown away, right? And in particular, a lot of input is thrown away that is self-generated. Right? So all of you probably don't feel the touch of your clothes on your body, right? Or you don't think that the world has changed just because you've moved your head, right? Or Daniel Volpert has taken that to the point where so the idea that you discard predictable information or self-exerted sensory data sort of stops you from being able to tickle yourself, right? So the underlying idea is basically that there's the world out there that changes, and there's us who change, who move, right? And when we move, we change the sensory data that we receive, but there's also actual sensory changes out there in the world. And the thing that we're interested in is the actual changes out there in the world. And the way to infer them from these sort of wildly changing sensory data would be to say that, well, if we issue a motor command, if we move, then we should predict what the sort of commensurate change in the sensory data should be. And then we should look at the actual data, and we should take the difference between those two, because that's the stuff that we can't account for by what we do, okay? And this is then basically something like a prediction error. And these prediction errors should relate to the things that are actually going on out there in the world. And so this is something that is sort of conceptually, I think, and something very intriguing and convincing, but it has this element in here, which is this minus sign. Right? So you should take the difference between actual data and predicted data. And whenever you see a difference and you think about networks that are Dalian, you immediately think of excitation and inhibition. Right? And so there's recent very beautiful work from the lab of Georg Keller, who's basically taken exactly that stance and is starting to unravel experimentally the sort of the neural circuitry, the inhibitory circuitry that underlies the computation of these types of prediction errors. And so the system in which he's doing that is visual cortex of the rodent. And sort of in earlier work in 2012 or so, he has basically shown that there's a substantial fraction of neurons in visual cortex that respond to prediction errors. And the setting is the following. You have a mouse that's running on a wheel. And then you have a VR setup where you can sort of control the visual input that the mouse is receiving. And if you close the loop, then you can move that visual input at the same pace as the mouse is running. Right? So that's sort of the normal experience. But then in VR, you can also break that. Right? You could basically make these patterns stand still although you're running, or you could have a scenario where you basically have them moving while you are not running. Right? And so you have full control over that. And what Georg showed is that whatever percentage of about 20 to 25% of the cells in rodent V1 or mouse V1 react to negative prediction errors, to situations where you're running, but the visual images are not moving. Okay? And that's something that he calls feedback mismatch. And so this is one specific cell, and you see that that cell actually has a very strong deviation from baseline in that condition, but doesn't react very much in other conditions. And so what Doreen has basically done is to see if we can explain that type of thing by sort of an experience-dependent balance of excitation and inhibition. And at that point, we're finally moving up to 3 interneuron types. So the setting is the following. You have feed-forward sensory input that is impinging onto this sort of canonical interneuron circuit of excitatory cells, and 3 types of interneurons, PV, somatostatin, and VIP. And there are also feedback predictions, basically, that arrive within the same circuit. And so what Doreen basically then did is she made those synapses here that are marked with the star plastic, and plastic such that they're basically optimized for an excitation-inhibition balance. And what she shows then is that you robustly get out neurons that respond only to these types of mismatched signals. Right? So these neurons basically become error neurons. You can also check what the response patterns of the different interneurons are. And so I think the very intriguing thing that Doreen has basically found is that the activation patterns of these interneurons are actually not quite as informative as something else, namely targeted manipulations. And so this is now going very quickly, but this balance of excitation and inhibition now becomes a non-trivial task if you talk about all these interneurons, because the inhibition is now mediated through a variety of different pathways. Right? So let's say you have visual input that is arriving in the principal cell, that would be excitation. It's also arriving in the PV neurons, then it would be feed-forward inhibition. Right? You could balance those two, all right. But it could also be that sensory data arrive in the somatostatin, so visual data arrive in the somatostatin neurons, which would then inhibit the PVs, which would inhibit the PCs. So you would have a disinhibition by visual data. And so you can basically take this further and further up to sort of trisynaptic connections where VIP neurons go to some neurons, which go to PV, which go to PC, which is sort of a disinhibition. And so what Doreen has found is that if you optimize for balance in these circuits, you end up with a scenario where you have all these different pathways that basically arrange themselves such that the sum of these pathways basically generates a balance of excitation and inhibition. And so how exactly they do that depends on where these feedback and feed-forward signals arrive in that system. And the advantage of a model is that you can systematically play through all possible configurations there are, which is something obviously that you can't really do in an experiment, and might help to unravel where these inputs are actually going within that circuit. So she simulates optogenetic experiments to knock out particular cell types and find sort of unique fingerprints that you could check for certain circuit motifs and input configurations. But I think at that point, I will probably leave the rest to Doreen, because it's sort of it's very extensive work, and I don't think I can really give it credit here. within this relatively short period of time. So I'll wrap up here. Oh yeah, right. This is published. So if you want to read this, it's in eLife. It just appeared about a month ago or so, I think, or two. I don't know. Okay, so let me quickly summarize. So basically, what I've told you is sort of 3 stories that all circle around this idea of optimizing for excitation inhibition balance. If you do that, then you can basically kind of reproduce the microcircuitry in mouse V1. You can, by optimizing excitation inhibition balance in a compartment-specific way, you can basically generate cell classes, inhibitory cell classes that resemble those that we find in the brain. And finally, just briefly, that you can relate excitation and inhibition balance to prediction error coding in primary visual cortex. With that, I'm at the end of what I want to tell you. Of course, it's important to thank everybody, that is the funding sources, but of course, first and foremost, all the people who actually did the work. This is a picture of the lab that is kind of a little bit outdated. I didn't have the time to put in a more recent one, or to sort of make it as beautiful as it should be. And specifically, of course, the people who did this work, which are Doreen, Joram, Owen, and Laura, and Richard, who contributed quite a bit during the time when he was here. And of course, thank all of you very much for the attention and for your patience for this pretty long talk. Thanks a lot.
Congratulations. Thank you very much. Or that I shall simulate by my voice. We already have 2 questions to be asked by Peter Jonas, who joined us on screen. So, Peter.
Thank you very much for the nice talk. I had 2 questions. First question, does the inhibitory plasticity, to the first part of your talk, involve changes in conductance or changes in ECL or changes in kinetics? What exactly do you have to assume as a plasticity mechanism?
Quite frankly, I think we're agnostic to that. We don't really know. I mean, the model is a rate model, right? So in that sense, the transmission is really sort of, it's a very impoverished perspective of just saying there's a single number, which is a synaptic weight, that tells you how strong the transmission is. So I think at this point, we don't really know what exact mechanism is. Of course, there's all sorts of interesting parts there, right? Because in principle, one could think about this being release probability, which would then change short-term plasticities, things, etc. But that's all stuff that I think we would probably have to leave up for grabs for future work.
And the second question, very briefly, you have talked about dendritic inhibitory cells and perisomatic inhibitory cells. If you would add bi-stratified cells with intermediate properties, how would that change the picture?
Yeah, I mean, that's something, quite frankly, that would be great, right, if we could get more cells. That's something where we don't exactly know how to do that. And I think in a way, when we do this modeling, where we try to get those 2 cell types from something like an optimization, then how many cell types we will get will probably depend on how many sort of constraints need to be satisfied. Right? So in this situation, you would need to satisfy an EI balance constraint on the dendrite and on the soma, and those cells are 2 compartment neurons. So yes, well, you get 2 types of cells, right?
Yes.
So in a way, you could imagine that if you'd move to more complicated settings, right, where the pyramidal cells are sort of more complicated devices, that you get more. But I can only very vaguely speculate here on what would come out if you would go there.
So you would need a detailed cable model for addressing this question, obviously.
Yeah, one could try that, but I mean, I mean that's not my home turf, right? It would be great to check that, but I'm not smart enough to make predictions there. What exactly would happen in that case?
Thank you very much.
Good. Thank you.
All right, Henning. One question of more general nature as to whether you can suggest any papers on research on delayed gratification that are connected to inhibitory synaptic plasticity. It's a bit left field, so I don't know the answer myself, but if you have any suggestions.
No, we can, I think we have to take that offline. I mean, I'm not delayed gratification by related to interneuron plasticity.
Yes.
Oh.
Yeah, I drew a blank myself, but I didn't want to not ask the question. In any case,
you can tell me what the background of that question is at some other point.
Well, no, it came, and maybe the asker will come into the Zoom chat. I wanted to say before, oh, we have another question. Hold on. Does the generation of classes, soma targeting and dendrite targeting, survive even when you introduce interaction between them, self-inhibition? Will these connections become reciprocal?
So, I mean, that's the question of what the interneuron connectivity is, right? So what I haven't really shown you about, and we're looking at that in detail, is that these connections are actually there. And so we're optimizing the connections among the interneurons as well. And so what the tendency is here that we get, I mean, you can do math on that and do all sorts of stuff. But basically what we get is something that is more on the bidirectional thing. Actually, there's a tendency in the opposite direction of the thing that, for example, Pfeffer and Scanziani or so have suggested. So you get more of a pronounced connection from the PV interneurons onto the SOMs rather than the other way around. And so the reason for that is basically that short-term plasticity model of Henry Markram and Misha Zodeks has a limited capacity of burst detection. It's simply because background activity will always change the release probability. So you can never, in the working of the system, get a setting where the first spike does not generate a response. Right? So you basically always get a response to the first spike, and then more for the second, but you always get the first. And so the problem is that if you want to detect a burst, then you have to discard that first spike. And the most efficient way of doing that is having feed-forward inhibition through a system that depresses, right? That measures that first spike. And that system, that is there in that system. That's what the PV interneurons are doing. So in that sense, this projection is something that we find quite prominently, but we don't know exactly whether this is sort of a consequence of the limited capacity or the limited power of the Markram-Zodeks model, and whether this would be something that would basically go away if you move to the point where we'd have a synapse that is facilitating to the extreme, right? Which is actually documented in the literature, that the first spike really doesn't do anything, and that it's really only the second spike that generates something. That's something that's virtually impossible to get with the Markram-Zodeks model. Did that answer the question? I mean, we did a lot more stuff in terms of knocking out connections to see which ones are relevant, etc. We're preparing the paper right now. If you want to learn more about that, feel free to talk to Joram. He's presenting this work at the Bernstein conference next week. So that's one opportunity. Otherwise, you'll have to wait for the preprint.
I also wanted to say, if you want to know more about this, join us now in the Zoom room for post-talk chatter. This was suggested by someone last week in the audience. They said, why can't we meet with the speaker after the talk? So Henning, I would like to join us too, of course. There's a link on the right-hand side. We'll see if we get involved. I doubt it, at least it look like it. And I'll see you all there. Before I go, I wanted to quickly make a plug for tonight's Learning Salon with Joshua Vogelstein and Tony Zader, leading a discussion about the nature and information content of the genome with regard to the architecture of the brain. Henning, can you mute yourself?
I can mute myself, yeah.
And then I wanted to make a plug for next week's Neurotheory Forum. Venkat Ramaswamy is going to talk at the same time and same place here. I hope to see you there, and I will see you all in the Zoom room now. So Henning, I'll unmute you. You can say goodbye, and then I'll...
Good. Then I guess I'd unmute myself. Yeah, thank you very much. Sorry, as you did that already. Thanks a lot, everybody, again. That was fun. And yeah, if you have any more questions, just join me in the other one. And I guess have a nice day.
See you there. All right.